thank you for being here, and possibly for other viewers who may watch this later on. Today is one of the tech talks for Rocket Reloaded, for one of the projects within Rocket Reloaded, and specifically the Pure Forensic DNA Profiles project by IFG and VyCAP. And this is one of the projects that is funded through an Interreg project. And I'll just briefly share my screen to provide you with a bit of an introduction of the context where this is taking place. You should be able to see my presentation right now. Like I said, this is one of the innovation projects within the framework of Rocket Reloaded. And Rocket Reloaded is an Interreg project funded by Interreg Deutschland Nederland, a European source for funding, and focused on, in this case, two main priorities. On the one hand, stimulating innovation within the cross-border region for the Netherlands and Germany, and on the other hand, promoting collaboration and getting to know each other, building partnerships sustainable for a longer period of time. So not just for this project, but hopefully continuing as we move forward. And to just briefly sketch the context, Rocket is an abbreviation for regional collaboration on key enabling technologies, such as photonics, nanotechnology, micro-nanoelectronics, etc. So a bit of technological development, but always resulting in new products or new services. Yeah, and within this framework, there are 6 innovation projects, each one between 275 and 400,000 euros. In total, 26 companies have participated and have received financial support. And Rocket Reload is a follow-up. We had a previous program before this, Rocket, which was a bit bigger. And within that project, there was some European recognition that these types of formulas, instruments, projects are best practice to really help stimulate these developments within Europe, because it helps companies to think outside of the priorities for today and focus on the middle to long term and see what is happening and what partners throughout Europe, and in this case across the border, could be interesting for them to help with that, to support that, to bring in new knowledge. So the framework is activated by 4 partners in this case. We are the lead partner from OostNL, the regional development agency for the Eastern Netherlands, and we do it in close collaboration with NMWP, the Nano Micro Werkstof Photonica cluster from North Rhine-Westphalia, Holland Semiconductors, and the TAF, the Transferagentuur Fachhochschule Münster. Just a brief overview of some of the results. So these are some of the figures that are relevant. We've helped within Rocket Reloaded 52 companies, of which we supported 26 financially, and we expect that there will be 48 new jobs because of this in the coming 5 years. One thing to note, a general invitation to everybody watching, coming Tuesday is the end event for Rocket Reloaded, because this project as a whole and the 6 innovation projects within it are nearing its end. So on the 17th of May in Ulft, near the border, we'll have an event where all innovation projects show what they have accomplished, what they achieved, and what they will be doing next, as well as some information on the follow-up program Interreg 6, which may provide new funding opportunities for them. So we hope to see everybody there. That's it from my side. And Mr. Hohoff, I think the floor is yours to show us what you and Kiki have done within this project.
Yes, so I would also agree. So I would also give the floor to Dr. Carsten Hohoff from IFG Münster to tell us more about the output and the results of the Pure Forensic DNA Profile project. So the stage is yours.
Yeah, thank you for the very kind introduction. The next slide, please. So to briefly sum up the project partners, so IFG and VyCAP are 2 small medium enterprises across the border. The IFG is an expert in forensic genetics. We are offering services and products for the forensic market. And VyCAP is an expert in single cell isolation, and they are offering products to isolate single cells for DNA analysis. And the goal of our project is to convert so-called mixed forensic stains into pure DNA profiles by application of the VyCAP technology, the Puncture that produces single cells. So next slide, please. As you can see here at the left-hand side, this is VyCAP based in Enschede, and on the right-hand side, the IFG Institute of Forensic Genetics in Münster. So some 70 kilometers separated. VyCAP is a spin-off of the Twente University in Enschede, was founded in 2011, has 8 employees, and it offers unique solutions to enumerate, isolate, and analyze rare single cells. It was invented to determine circulating tumor cells. There are several tools for single cell isolation and characterization, device disposables, and we will have a closer look later on. So next slide, please. The IFG is a Münster University spin-off founded in 2007 by Professor Brinkmann, the head of the Institute of Legal Medicine, which is something particular in the German-speaking countries. They have the discipline called legal medicine. The IFG has about 15 employees. Some of them have more than 20 years of experience in forensic DNA analyses. And the unique selling point of the IFG is that we are offering the so-called JETNEP proficiency test. This is a blind trial used to qualify forensic laboratories worldwide. The other items that IFG is offering, these are forensic DNA analyses in stain cases, in parentage testing, and in the cell line verification. Next slide, please. Here's a brief overview of the JETNEP proficiency test. There are more than 200 participants from more than 45 different countries, clustered mainly in Western Eurasia, of course, in Europe, but there are also participants from North America, South America, and Australia. There are 3 groups of laboratories: university labs, state labs, and private labs. And this is also important then for the market access for the project later on. Next slide, please. The classical portfolio of the IFG is, as we can see here, the stain analysis. We are offering this to the German law enforcement agencies, prosecutors, courts, police officers, and so on. And the main issue is analyzing DNA by the so-called STR typing approach. STRs are mini microsatellites that are good only for the DNA typing. And then later on, the IFG experts defend the results of the forensic DNA analysis then in court. So next slide, please. Here's an overview of how forensic DNA typing works. Next slide, please. It's a mixture of biology, technology, and genetics. The key issues are DNA extraction, DNA quantitation, and then the PCR-based amplification in a multiplex format of those STR markers. DNA extractions in many disciplines is not an issue at all, but in forensics, the DNA might be compromised, it might be degraded, heavily changed by environmental impacts. So it might not be in a proper condition. The technology issue is the separation and detection of the PCR products. This is done by proper length measurement of those STR alleles. And those measurements are then converted into internationally accepted so-called genotypes. And then the genetic part is that it makes sense to compare the result of a sample or a forensic stain to a person, a suspect or a victim. And if that is not available, then there are so-called DNA databases in all European countries, and you can compare a result of a stain to such a national DNA database. Next slide, please. Again, all those steps that are shown on the left-hand side, this is a crime stain, and on the right-hand side, this is how suspect is analyzed. It's always a combination of several steps, including collection, sample storage, sample manipulation, then the DNA extraction step, quantitation, amplification, separation and detection issues, data interpretation, that is also interesting, and then a statistical interpretation. So is it an exclusion or inclusion? And if it is an inclusion, then of course, the resulting figure is important for jurisdiction. And at the end, a report with a statistical weight is produced by the expert. Next slide, please. Here you can see a typical electropherogram in the right-hand side. This is a STR profile, at least 4 different profiles in rows. And on the left-hand side, there is a schematic drawing of what can be expected. So if you have a so-called pure sample, single source sample, which is derived from a single person, then you only observe 1 allele in a so-called homozygote status, or 2 alleles in a so-called heterozygote status. But this DNA can be mixed, can be a mixture of several persons, and then we have a so-called mixed stain. And next slide, please. Those mixtures, here an example on the right-hand side, those mixtures are much more difficult to interpret. And while on the left-hand side, it's very clear if you have a match between such a pure profile and a suspect, then you can easily calculate high figures. So such a DNA profile is unique in the universe, while a mixture is very hard to compare to a certain person. Next slide, please. And for the jurisdiction, a single source stain, it's a lot more convincing. The numbers, the likelihood ratios are very convincing, very high, as I said. While in mixtures, in mixed stains, you sometimes have to calculate in a different manner. You can only use this random man not excluded approach. And sometimes you end up in figures such as 1 in 1,000. And thus for court, the evidential value is limited. And a court might not be sure that the person is indeed the donor of the stain or the co-donor. And therefore, next slide, please. We have thought about, would it be possible then to convert such a mixture into pure DNA profiles by separating those components of a mixed stain to separate cells that are in this stain and to type those cells separately to generate single cell DNA profiles. And then we got the chance to meet VyCAP, to talk to the experts, and to have this project together. VyCAP is an expert, especially in blood cells, in white blood cells, so-called leukocytes. Forensically, blood is very important, but there are other cells as well, sperm cells, skin cells, that might be in a mixture, sometimes in combination with blood. Sometimes you have a sperm cell mixture, or a sperm cell vaginal epithelial cell mixture. Next slide, please. An issue is the status of a sample. In clinics, you have usually a liquid blood sample. In forensics, a sample is called usually a stain, and this stain is usually dried when police officers find the stain. And as I said before, such a sample might be compromised, so DNA might be degraded and contaminated with other components, or sometimes heavy metals, dust, whatsoever. So whatever is outside, carbohydrates, proteins, lipids. So isolation of the DNA is critical. You have to separate the nucleic acid from other cell types and components of such a stain. Sometimes you have the issue that there are cells of non-human origin present as well. Some of us have pets, so cats and dog DNA might be an issue. And very interesting in the forensic part are the so-called shed skin particles, sometimes called corneocytes. We are losing 400,000 of them each day. So even perpetrators do at the scene of crime. And forensically, they are very, very interesting. They are composed of several cells. So the particles are a lot larger than, for example, a leukocyte. Next slide, please. So here you can see a schematic drawing of the forensic workflow that we have established. On the left-hand side, you have the forensic item, maybe a knife with a red brownish fluid on the blood. Then we have to prepare the sample to make a proper cell suspension. The next step that VyCAP has developed is a so-called pump unit and a disposable part which can contain a certain slide with a chip, which I can come on later on. Then in the middle, we have the Puncture system, which is also a unique selling point. And this Puncture system allows for cell separation. So cells can be transferred to tubes. Within the tubes, cells can be lysed, DNA can be deliberated, and this DNA can be amplified with classical forensic genetic techniques to produce an STR DNA profile shown on the right-hand side. Okay, next slide, please. And the left-hand side, you see the consumable devices, and in the middle, we have a video that shows the principle. It is a microchip. Above, you can see those floating cells, those leukocytes, and each well has a pore. So liquid is passing through the pore until a cell enters a well, and it blocks the pore. So the principle leads to the situation that within every well, there is one single cell. And if you have a good image capturing device, then you can also have a look and demonstrate. Next slide, please. Now we have a video of the Puncture Pro, which is the device that does many, many steps automatically, as you can see here. There is a certain software that allows manually or automatic selection. We have the possibility to label the cells. Forensically, as I said, there are leukocytes from blood, there are epithelial cells, there are semen cells, and for all of them, there are particular antibodies that can be labeled fluorescently, and then you can characterize a cell that is bound in the cell, in the microwell. And whatever the question in the forensic case is, you then can select such a well and the containing cell and proceed by punching. Next slide, please. The Puncture system is also a unique selling point. By punching, you can transfer the cell of interest into a PCR device. But starting the process on the left-hand side, preparing a cell solution, cell suspension, then from a forensic sample, a proper labeling, as I said, to be able to determine leukocytes, epithelial cells, semen cells, then a particular pump unit in the middle, and a single cell disposable and a slide with this microwell chip. Next slide, please. So here you see the principle of the chip, which is made from silicon. It has 6,500 microwells. The volume of each and every well is very small, 2.5 nanoliter. The bottom is transparent, so that's good for microscopy. And as I said before, each well contains a single pore for liquid transfer. So the size is optimized for leukocytes. Next slide, please. And here you can see the high-resolution single-cell imaging. You can also detect several fluorescence dyes, leading to a proper characterization of the cell that is within this well. So you can distinguish between blood cells, white blood cells, semen cells, and epithelial cells. Next slide, please. And then the puncture needle is used. This puncture transfers the bottom of the tube, which is also made from glass, into the PCR tube. It is inert, it will not interfere with the PCR reaction. And this is the chance then to transfer the cell of interest into a PCR tube. And then you also have a good control by microscopy. We will see pictures of that. And it can be rather fast. We were talking about automation. It is possible then to automate it, that a single cell per second can be punched and then transferred to a PCR tube, as you can see in the middle. And the design of the needle is properly made, so it won't touch a cell during the punching process. So next slide, please. Again, the workflow. On the left-hand side, the IFG is producing stains. VyCAP has developed a protocol to convert those stains into a proper cell suspension. Using the pump unit and the microwell chip, cells are captured and separated, and the puncher then provides the automated fluorescent imaging, selection and isolation of white blood cells, semen cells, and epithelial cells. And then we have single cells for the STR analysis. The steps in the middle, they were done with VyCAP, and IFG is doing the STR analysis that you can see on the right-hand side, the so-called single cell STR, or sometimes we can pool certain cells, 5 cells, 10 cells, to estimate if there is a critical mixture and to estimate the mixture ratio. Okay, next slide, please. Yeah, in forensics, the sample type is critical and of interest as well. There are several so-called swab types that police authorities use to handle such a forensic stain. There are nylon swabs from the Italian company Copan. Sometimes they are using viscose or sometimes cotton. They all have their differences. The nylon swabs have the advantage that they are completely done chemically. So there is no authentic DNA, no plant DNA inside. They can be made very pure with clean room circumstances. As you can see here in the middle, there are some images made by the Puncture system. At the left-hand side, the tube ID is shown and the well ID is shown. The next row, this is an image of after the punch. So if you do not see any fluorescence, you can be sure that the cell has been removed from this microwell and is contained by the PCR tube. Next row, the so-called DAPI stain. this is a nuclear DNA stain. You can be sure that you have a cell with a nucleus, and the nucleus contains the DNA. The next row is called a fluorescent marker PE, and the antibody detects a white blood cell. And then you have another row showing the reflection image and the bright field image. And on the right-hand side, you see many, many images. These are 19 wells that contain a single cell that was isolated and then converted from a stain into single cell PCR tubes. Okay, next slide, please. Here's a typical image of what is the result of a forensic DNA analysis. In these rows, you have the so-called STR markers that are distributed in the human genome, residing on different chromosomes. If you have, for example, in the sex-determining marker amelogenin only an X, this is row 4, then you know that the donor of such a stain is a female, which is in forensics very, very important because many, many victims of those crimes are women, and the majority of perpetrators is male. So you have also a very easy selection then on those samples that might be suited to find the perpetrator. And then you have rows showing the results of the STR markers. The next would be marker D3. You have the alleles 16 and 17. So this is a heterozygote sample, and you can easily compare with a person of interest with a DNA of profile of the person of interest, and then you can exclude or include by combining the results of those markers. In the IFG, we started first with fresh white blood cells that work very well. There are some issues regarding this low copy amplification because it's only a single cell. Usually the IFG is used to handle 100 cells, for example. So there are so-called dropout events in line 3, the so-called sample C1.2. There is an asterisk at the marker D10. This means that this marker did not give a result. This is typical for single cell analysis, and in jurisdiction, you need more than a single cell to solve such a case. Okay, next slide, please. Yeah, to sum up, what has been done in the project and the Rocket Reloaded project combination and collaboration of IFG and VyCAP. Several protocols had to be developed, and successfully we have developed a protocol for rehydrating a forensic blood stain which contains dried leukocytes. So rehydrating is essential then to allow those cells to flow and to enter those wells on the chip. Also, small pool separation was possible and has the advantage of better signal-to-noise ratio. We were able to develop a protocol then to fluorescently label different cells, leukocytes, epithelial cells, semen cells, and this shows a human specificity. So we can, by choosing a proper antibody separate from, for example, PET cells that might be present in such a forensic sample. Next thing we were able to, that was to lyse those single cells and small pool PCR prior to PCR, and then we were able to optimize the amplification and detection of the PCR products by the so-called capillary gel electrophoresis, the standard technique in forensic genetics. Next slide, please. And we are in the collaboration optimizing other protocols for other forensic samples. As we have shown, we particularly specialized on blood mixtures, but saliva and sperm are very well or very interesting stains in the forensic fields. And I mentioned before, those shed skin cells, those corneocytes, which comprise of several cells that have been cornified during the skin maturation, are very essential. You might all know them from the scalp, those cells, and we lose so many of them that it is very wise to analyze them and to produce a technique, a protocol that can work with those skin cells. They are a bit more difficult than fresh cells like leukocytes. The DNA is degraded because the cells died 3 weeks ago in the skin and then were shed. But we are very optimistic when we can devise a protocol to use those shed skin epithelial cells. And the next step that we are working on, these are mixtures of blood, saliva, blood, semen and saliva. These are very often found mixtures in the forensic scenario. Okay, next slide, please. And the outlook, we intend to publish those results, also to be present on certain forensic meetings. That was very sad in the last 2 years due to the COVID-19 pandemic. There were no meetings, scientific meetings in person. And we had the first contacts. As I said, we have a very good network with other forensic institutes worldwide. Kiki Andre made a presentation of during the Spuren workshop, a stain workshop in Germany, and we were contacted by putative customers, other forensic institutes, and police authorities. And the first order has been received to separate cells in the case and then to do the DNA typing. Okay, next slide, please. Other markers are of interest, markers for pigmentation analysis, so to determine the color of the eye, of the hair, and of the skin is allowed in Germany. In Netherlands, it has been already been allowed for many years. In Germany, it started in 2019. It is of interest to the forensic community as well to type single cells. And what is also allowed is a so-called age estimation. In forensics, it might be of interest to determine the age of a donor, of a stain, or of a cell. Maybe if you do a so-called mass screening, then it's wise to have an age estimation of the donor as well. And one might think of a so-called true walk-away automation of punching, like doing with artificial intelligence, with a certain setup then that allows for collecting of the cells of interest. And we are thinking of a lab-on-a-chip approach for integrated amplification and detection of those single cells. Next slide, please. And of course, I would not like to stop without thanking the collaborators. On the VyCAP side, in addition to Kiki Andre, Lisa Omens, Yvette Peters, and Joska Brugmaat, and the IFG, Kirsten and Miriam were involved in the project. And on the Rocket Reloaded team, there are, amongst others, Julia Neuenhaus, Johanna Jürgens, and Alex van Gelkop. So thank you very much for your input to this project. And thanks for listening, for the audience. Thank you for your attention.
Yeah, thank you very much for this interesting talk about the outcome of the project. I think you mentioned it already a bit, but I thought about now. I think goal is that you of the project was to identify single cells, but I think now you could think otherwise. What about automation to identify single cells, but 1,000 single cells at the same time? So I think in this case, possibly do you, or my question would be, do you separate and identify the cells by hand, don't you?
Yes, in the pilot phase, we do it manually, but the software that the Puncture Pro has is also capable of doing some automation step. So Kiki is in the audience. Kiki, maybe you can add some comment on that.
Yes, I have. So what we currently can do is, because we identify the cells based on the fluorescent markers that we use to label them, and using our Puncture Pro system with the software that we have, we can actually do part of it automated. So we can make a selection on certain parameters. You can say, okay, I want to identify cells which have this amount of fluorescent, and in that way we can do kind of an automated selection. But in the end, always the operator has to decide which is actually a self-interest, or which cell you actually would like to isolate for further analysis.
Okay, but I think if you, for example, combine, because it's fluorescence, photonics with artificial intelligence, you could say in the end, okay, we are absolutely clear that no females were involved, for example. So you can say, okay, please system, please extract just male cells. The other ones are not interesting, or we're searching for blood cells or other cells. So if you can give your machine a certain kind of briefing, so it's okay, please differentiate. So in this case, the automation would do a lot of work that has to be done by hand now. That's right.
Yeah, that's essential in forensics. There's always a bottleneck. We have many more samples than capacity to analyze. So it's an advantage to have an automation and to be faster, and then to solve crime. That's at the end our task.
Yeah, I think in this case, for example, artificial intelligence is sometimes a risk as well as a chance, because, for example, if you in cancer detection, the picture recognition of artificial intelligence sometimes is more precise than some professors are. But on the other hand, we still need to have the capability to do this by hand at the end. But I think possibly, if you have an artificial intelligence and a good camera, that this might do a lot of work, because in the end, you need single cells to have this pure forensic DNA, as the project also was named. But I think the more cells you can handle at the same times, the better, isn't it?
Yes, but the bottleneck is afterwards. As I said, the chip has a capacity to separate 6,400 cells. The sequencer does not have the capacity to analyze so many cells at a certain time. Therefore, it's also worth to think about improving this step, the final step.
Okay. So this could be the future of the project, or the next step, for example?
Definitely.
What other next steps would you see?
Yeah, so we are thinking of offering services and products to the forensic community. Of course, we would like to sell machines, the Puncture Pro. At least in Germany, there are about 80 institutes that are doing forensic DNA research. Some of them are so small, they may not have the budget to buy such a device. Therefore, it is worthwhile of thinking that the IFG is offering those services to the at least German-speaking forensic community.
Alex, you have a question.
Yeah, thank you for the presentation, Karsten, and Kiki, of course. Very interesting to see what happened and how you did it. I was just wondering about the size of the problem. And mixed samples of DNA, I can imagine that it's something that happens all the time. I can also imagine that it might occur occasionally, because there most often will be pure DNA samples. So just wondering, is this a question that comes along a few times a year, or is this something that's actually, and if that's the case, it's still very relevant because any crime solved helps and is beneficial, of course, but, or is this something that happens in daily practice, for example? So how big is the problem?
It is big. It is big. It is also due to the increasing sensitivity of those techniques. So maybe 10, 20 years ago, we would not have detected those mixed stains. Now with the lower limit of detection, we find that a pure stain might be also composed of other cellular components. And there's one big group of samples, they are so-called touch samples, like door handles. And these are really, you can expect many so-called mixed stains. And nowadays, one approach is to do a calculation step, to separate mathematically by some means. But more convincing in court is indeed to separate the cellular components, to characterize also the cellular component, to be able to say this DNA profile is found in a leukocyte or in the epithelial cells. So this is an extra information that we can provide to the jurisdiction.
Interesting. So it also helps, even though cases might be already being processed, but it could help to increase the believability and of the samples in court.
Exactly, yeah. And it's also suitable for so-called cold cases. The DNA is stable for many decades, even centuries. And this is also an approach to solve those old cases.
Oh, nice. Okay, interesting. Thank you.
Yeah, I would have another question. Have you also spent already some thoughts about how to pick up those single dead skin cells? I don't know the name. I can't remember it, but those you lose all the time.
Yes, we have spent some ideas on that. The difference is they are not that soluble in water because they have a high lipid content, and therefore we think of adding some detergents to the solution to make them better solvable, or in a solution or in a suspension format. The main issue is the size, the pure size. It has a size distribution, of course. The majority might be too large for the wells that we use. And so we think of adapting and changing the microchip as well. But it's more difficult than other aspects of the workflow.
Yeah, I think over that, how do you pick them up on the crime scene with a vacuum cleaner? And if you would think about this, you would have, the complete dirt in your probe. So it's even harder to differentiate. So in this case, again, automation would be the key, because you would have a lot of, dirt and just some single cells in this. Okay. I think, yes, one of the associated partners is very interesting because it's the Dutch police who said that this is a very interesting project. Are you still in contact with them? What do they think about the output? And maybe the second question, what about the COVID pandemic? Made it your progress more difficult, and also the contact to partners like associated partners who are not so involved into all those processes?
So maybe Kiki, can you answer the aspect, the forensic Dutch police?
Yeah, definitely. Yes. So throughout the project, we had quite some contact also with the Dutch police, who were also interested in this in this whole project. And I think also within the project, at one point we started a small collaboration with the Saxion University of Applied Sciences, where the Dutch police also is involved in one of the research groups there, looking into forensics, new forensic technologies. So we started a small collaborations where they tried to do similar things, also look at signal cells, get signal cells from forensic stains. They were very interested in as well, and I think the Dutch police still is. So they, we updated them throughout the project, and they were very positive also about this whole project. Yeah.
Okay, thank you. One follow-up question. After that, René, you can ask your question. But one question from my side would be, what about, do you need for these types of machines, do you need approval processes before you bring them on the market, before you sell them? I think so. Or not?
Yes, in the forensics, you have to do a manufacturer validation to show that they are fit for purpose. And then in addition, the laboratory that has bought such a machine must do an additional validation step, so to demonstrate that they are capable of using this machine properly. So there are several steps then to make it well in the jurisdiction process.
Okay, but the validation of the users in the end, I think you can help with this, can't you?
Yes.
Okay. Okay, now, René, you had a question as well.
Yeah, thanks also from my side for this very interesting talk. I was wondering, you mentioned publication, which is in process at the end. I can't imagine that you will publish all the stuff, how the machine operates and how you do all this diagnostic. So I think it's more like the scientific approach, which will be covered in this publication, but maybe you can give a little bit more information what's exactly to expect it when reading this publication.
Yes, exactly. This is planned to show the scientific summary. The forensic field is always interested in something like success rates, problems like dropout that we mentioned. And it's, in addition, it's a kind of marketing for us to be able to demonstrate, to publish that the workflow works. And this is a good marketing argument as well.
Thank you.
Okay, thank you very much. Do you have further questions right now?
Perhaps one final one from me, if it's possible. I was just wondering, you've mentioned some of the steps that you're going to take towards the future. How do you envision that, if that remains, that your collaboration between IFG and VyCAP will continue? Is it in the development phase, or do you think it would be somewhat of a service provider and that IFG has the questions and the samples that need to be analyzed, and that VyCAP does the service, or do you provide the machines? Do you have any thoughts on that? If you think that the collaboration will continue after the project, of course.
Well, to be honest, there has been some changes in the IFG. So the managing director has been changed, and talks are necessary then between the new managing of the IFG and VyCAP. So I have a personal point of view, of course. I would really be more than happy to go on with the collaboration. It was very fruitful, but now the new management has to decide on that.
Yeah. Okay. Clear. That's always something to wait what somebody new thinks about it. So okay. Thank you.
Thank you very much. Further questions? Yeah, if not, I think I will send around the presentation afterwards, or at least the video, as soon as I got the permission for this from Dr. Hohoff. So from my side, I would again thank you all for your participation. I think it is a very interesting project, a lot of things were achieved, and I think a lot of things will come in the future. I see it like this, and I wish you good luck for the future. And again, as Alex already mentioned, I think we all will meet next Tuesday in Ulft for the Rocket Reloaded End event, and I think then we can continue our discussion in personal. Thank you very much.
Thank you.
Bye-bye. Thank you.
Thank you.
Thank you. Bye.
Thank you. Bye.